Hey boys and girls, I wanted to take a couple of minutes to just go over the PowerPoint again. This will be especially helpful for those of you who weren't here and for those of you who found the content a little confusing, because we're definitely getting into some more messy concepts than we've gone through in the past. So as we said yesterday, the diffusion is the movement of molecules from an area of high concentration to an area of low concentration. So it could be in the air, it could be in water, and we talked about all the different examples. But basically, if you have some sort of air freshener and you spray it, it's going to smell a lot stronger right near where you sprayed it. But eventually, those molecules are going to want to spread out so that they're even. Because remember, nature in general wants to create equilibrium. I know that we talk about homeostasis in the body, but nature wants things to be equal. So here's an example of diffusion. So dropping food coloring in water. And then here is another example. You can read the examples like up here. So we talk about muddy water. So when you have mud and you mix it with clean water, you can see that the mud kind of just makes a muddy mess of the clean water. Any of you who put, if you have coffee or you see your parents drinking coffee and they pour the creamer in, happens to me every morning. When I put my cream in, it kind of centralizes in one place, but then eventually it spreads out. So here we have tea. Put a tea bag in water. The water starts out obviously completely clear, but then eventually all of those molecules of tea spread out so that you don't, with every sip, you have the same amount of tea, and it's not water in one and then tea in another. And then, again, here's an example of some candles. So it starts to get a little bit more complicated when we talk about osmosis. So osmosis is diffusion, but it's the diffusion of water, okay? That's really important. So anytime we're talking about osmosis, we are not talking about any other molecule other than water, okay? Water gets its own process, and it's called osmosis. So the two differences between osmosis and diffusion is that it's water and it requires a semi-permeable membrane, okay? So when you are spraying air freshener, the air freshener doesn't actually have to cross through a membrane to diffuse around the room. It just goes in the air. Osmosis, in order for it to be called osmosis, it has to cross a membrane. So when you're taking a shower and the water is really hot and it starts to make steam, that's water. And then it starts to fog up the mirrors and you can kind of see it in the room. That is not osmosis. That's the diffusion of water, but there's no membrane. So it's still called diffusion because there's no membrane involved. So this membrane is selectively permeable, and we've talked about that before, which means it allows some things to come in, but not everything. And that's usually dependent on the pores that are in that membrane. So they're really small, so the chances of a smaller molecule getting into and through a cell, through a membrane into a cell, are much greater if the molecule is really tiny, like water or iodine, which is what we used for that osmosis demo that we did yesterday in class. And I'm going to show you some pictures of that in a minute. So we just want you to have a kind of an overall understanding of these solutions that we've been talking about. So when we're talking about a hypertonic solution, this is where osmosis occurs, okay? So that's why we're bringing it up. We're not trying to throw in extra vocabulary, I promise. But osmosis occurs in these three types of solutions. So one is a hypertonic solution. So I know that these are the notes that you need to be taking. So make sure that you fill in your PowerPoint with this information. But this is the information that I want you to pay attention to right now because I think it's going to help you understand what's going on. So in a hypertonic solution, remember hyper means too much or high, okay? So The solution is the water or whatever that is in the beaker that you drop the cell into. Okay, so we're not talking about the cell, we're talking about the solution. The solution that the cell is placed in is considered hypertonic because it has more either salt or sugar, depending on what's in that solution, than what's inside the cell. So if there's more salt, there's less water, okay? So if there's less water, that solution is going to be like, hey, I want that water. So it pulls it out of the cell and makes the cell shrink and look something like that. So remember, you drop a red blood cell into a hypertonic solution. It means there's a lot of salt. That means there isn't as much water, okay? And so the solution will pull the water out of that cell and make that cell shrink. So something that I told my students yesterday was, I want you to remember that salt sucks. Now, that is not normally a word I want you to use, but in this case, we are talking about the action of actually sucking water out of something. So any of you have done any gardening, or if you're familiar with slugs, they're kind of a big fat snail without the shell. They get into your garden and they can make a mess of things. If you pour salt on them, which seems horrible, but if you pour salt on them, slugs that have a lot of water in them, it'll dry them out because it'll suck all the water out of it. So now we have the opposite type of solution. We have a hypotonic solution. So hypo rhymes with low. So, well, kind of. Sounds like low, which means there, and remember, this is the notes that you're putting in your PowerPoint. But this is the information I want you to look at right now. So the solution that you put the cell in, does not have very much salt or sugar in it. It actually has more water in it than what's in the cell. So the cell's like, hey, wait a minute, I want that water, and it'll pull the water in. So what happens is the cell starts to swell, and eventually it can burst, which is obviously also bad, right? So the cells burst, and then the organism can die. Isotonic is when there's the same amount of solute and water inside the cell as outside the cell. So this is the ideal, all right? This is the kind of solution that is given to you in an IV, or if you go into the hospital and you're just getting saline solution. That's what that's called, saline solution. It is going to have, or it needs to have, the exact same amount of solute and water in it as what's in your blood cells. Because if it had a salt water solution, then it would completely dehydrate you and kill you, and that would be bad. If it was distilled water or a high water concentration, it would make all of your cells explode. So that kind of defeats the purpose of going to the hospital, I would imagine. So here's an example. So this is what it looks like in an animal cell. So hypertonic means it's in a beaker with a lot of salt. That's the symbol for salt. So lots of salt, okay? So it's going to pull the water out of the cell, make it shrink. This is what it looks like in an animal cell. This is what it looks like in a plant cell. Notice that the cell wall is not changing. Okay, remember we talked about how it's actually the cytoplasm that shrinks. Okay? Isotonic, it's a beautiful little red blood cell, and that's a plant cell, and both of them are staying the same. Hypo means there's lots of water in the beaker that you plunk this cell in, okay? So what happens is the cell's like, hey, I want that, pulls it in, and then it makes it explode. So here you can see this is normal, and this is starting to expand. It kind of gives that cell wall a roundish shape, and that is not, so it says normal here. When it starts to expand like that, I'm sorry, eventually it's going to explode. So that's normal until the whole thing just blows out. So here are some other examples, and you can read through these. So isotonic stays the same. Hypertonic pulls water out of the cell. Hypotonic pulls water into the cell. So out of cell in hyper, into cell in hypo. Okay, so I did this demo of osmosis yesterday, and I have some pictures at the end of this presentation that I am going to show you that I took yesterday right after school and then this morning when I came in. So here's a lot of information. Again, it says to refresh your memory or if you're absent. So you can just read through this. I'm not going to go over this. Again, some more information for you. And more information. Okay, so now we get to passive transport. Passive transport is the overall, it's like the umbrella term. Let me see if I can draw an umbrella here. Ooh, look at that. For diffusion and osmosis. It's called passive transport because it happens automatically and no energy is required for it to happen. Okay, so, and what we mean by energy, we'll explain that in a minute. So, again, passive transport, the examples of passive transport are diffusion and osmosis. Happens automatically because of the body's desire to be in homeostasis. Active transport happens when, let's say, there is more water. There's a lot of water outside the cell, not as much inside the cell, but for some reason, that water needs to get pulled out of the cell, or whatever it is, needs to get pulled out of the cell. That isn't going to happen automatically because it's not going from high to low, it's going from low to high. So if it's going from low to high, that it doesn't make sense to the cell really, and it requires this transport protein that is like a little shuttle bus that like shoots it across the cell membrane. And whether or not that needs to happen is all dictated by the nucleus. So, two types of active transport. There are transport proteins, and like I said, it's like a little shuttle bus, and it picks up the molecules on one side and puts it onto the other side. So you can see here, this is the direction that the energy is moving. The molecules are moving. But look, there's very few here. There's a lot here. So why does this have to go this way? Well, it could be a lot of reasons, and we'll get into those details in a little bit. But just remember that if it's going from low to high, it requires energy. So it's called these transport proteins. And like I said, they're like little shuttle buses. They pick up the molecules and shoot them out of the cell, but it requires energy, which is it's adenosine triphosphate, one of those long terms that we have in science that fortunately we have an acronym for that. So it's adenosine triphosphate, but we call it ATP, and that is cell energy. Okay, so that's what's required with transport proteins. Okay. Another method of active transport is engulfing. So an amoeba, this is how it eats. It goes up to something, a food source, it surrounds it, and just engulfs it into its body. And that's an example of active transport, transport by engulfing. So these are the pictures that I took yesterday at the end of the day. So this is second period. This is third period, fifth period, and seventh period. So you can see, here's the tubing, okay? You can see that the tubing is getting lighter and lighter as the day is going on. So this was first, obviously second, as the day went on. So this was it. I took this picture at like 9:30 a.m. and I took this picture at probably 3:45 p.m. So you can see that the molecules, the iodine molecules, are going from the cell, and remember that's what we're talking about there. We're using this dialysis tubing to simulate a cell. The iodine is going into the starch solution, okay? So just to refresh your memory of what was in the solution. So to refresh your memory of what was in the solution, we had cornstarch. So this is cornstarch mixed with water, and then this is iodine. Okay, so cornstarch molecules are very big. Okay, iodine molecules are small. So what happened was we had this dialysis tubing, all right, and that is to simulate a cell, because dialysis tubing is used for patients that have kidney disease and they need to have their blood filtered artificially. So the blood actually goes into this dialysis tubing, and then the bad stuff comes out, and then the blood goes back into their bodies. But anyway, so here's this dialysis tubing, and the iodine went out into the cornstarch, okay, and turned it purple. Okay, so you can see that this is what it started looking like, and this is what it ended up looking like, because iodine is an indicator for starch. So it'll turn starch, or it'll turn purple in the presence of starch. So I guess you could say that it turned the starch purple, but actually the iodine leaked out and turned purple, then mixed with the starch, and that's why it's purple. So I took this picture this morning. So again, second period, third period, fifth period, seventh period. And look, this is completely clear, all right? So all of that iodine leached out into the cornstarch solution, and because it's purple, it mixed with the cornstarch and I guess gave it the appearance that the cornstarch turned purple, when actually it's just the purple iodine. So I hope this is helpful. Make sure you take your time with these concepts, and if you have any questions about them, don't just skip over it and assume that you'll figure it out down the road. You absolutely need to ask a neighbor, ask me, go to tutoring, watch the video again, read over your notes, but I really need you to have a solid understanding of this content so that when we move on to concepts that include this information, that you know what's going on. Make sure you ask questions.